Hey everybody, I'm going to go ahead and talk to you about a couple of different biochemical techniques. We're going to go through a total of 5 different techniques. We're going to talk about isoelectric focusing, southern blotting, northern blotting, western blotting, and an ELISA assay, or an enzyme-linked immunosorbent assay. So get ready and get pumped. And here we go. Northern blotting is the first technique that we're going to talk about. Northern blotting is a technique where you are going to study gene expression. What it detects is it detects RNA as well as mRNA. So it detects, in general, RNA molecules. It is not limited to mRNA, so it can detect short single-stranded, short double-stranded pieces of RNA. Now, what this is going to do is it's going to reveal the identity, number, activity, and size of a particular gene of interest. It was developed in 1977 at Stanford by James Allline, David Kemp, and George Stank. Now, this technique, one of the very first techniques, or first parts of this technique, is RNA extraction. So that RNA is extracted and is run on a gel electrophoresis gel. So this is an agarose gel. But one of the things that's very important here is this single-stranded RNA is very sensitive. There are RNases all over the place, and so it can break down or it can be hydrolyzed very easily. So there are some modifications that you have to make to this agarose gel. It's not the exact same as the DNA agarose gel because there are some things that you have to add to that gel. So what that ultimately is going to do, because you're doing electrophoresis, is these RNA molecules are going to be separated based on their size. Then what you're going to do after you run that onto a gel, you're going to transfer the bands from that RNA gel onto a membrane. Now that membrane is going to basically accept those RNA molecules, and then what you're going to do is you're going to incubate that membrane, not the original gel, but instead the RNA molecules transferred from the gel. You're going to incubate them with a labeled probe. So what you've done is your gene sequence, or part of it. What you're doing is you're basically saying, I know the gene sequence that I'm looking for, and so what I'm going to do is I'm going to synthesize a probe. This synthetic probe is what I'm going to incubate with my RNA gel. Okay. Now, what that means is if the gene that I'm trying to study and characterize is present in this RNA sample, well then my probe will bind to that, and boom, whenever I go to visualize my labeled RNA on an X-ray film, I will actually detect that. So this probe, chances are very strong that it is a radioactive probe. I say chances are very strong because of the fact that that RNA probe, current technologies enable you to use a fluorescent probe rather than, or some other type of label rather than a radioactive probe. So that's basically the rundown of what you're doing. Now, why would you actually do northern blotting? Well, one, you're going to analyze a sample of RNA, excuse me, to measure the expression of a gene. What it can be used for with respect to that, to kind of dive a little bit deeper, is it can be used to detect certain diseases and illnesses related to gene expression. So if you compare a population of people, and you have healthy peoples as well as diseased peoples in this mixture, or in this sample, what you could do is you can say, oh, well, as it turns out, there is this gene that is expressed only in, or sorry, this gene that is expressed only in diseased people, so maybe that has something to do with this disease that we're studying. Um, so southern blotting actually follows, or sorry, northern blotting, where we're probing for RNA, follows the development of the southern blot. And the southern blot was named after a guy named Edward Southern. We're going to be learning about southern blot after a little while, but the name northern blot is acknowledging that, and kind of it's just the play on words. So if you were thinking that it has to do with, oh, it was developed in the north or the south or something like that, it was not. It's instead a kind of play on somebody's name. Okay, so your technique basically follows: you isolate RNA, you run it on gel electrophoresis, you transfer that onto a blotting membrane. That blot where that membrane is exposed to, excuse me, sorry, a single-stranded DNA probe, and that probe is detected by chemiluminescence or autoradiography. So what I was talking about earlier with the radioactive probe, it would be autoradiography that you would be using there. Alternatively, like I said, you can use a probe that has a fluorescent label, which that would be your chemiluminescence. Again, there's kind of your rundown of that process. Okay, now what your results are going to look like. What we see here on the right hand side, this is an RNA gel. And so whenever you run an RNA gel on, or sorry, whenever you run an RNA sample on a RNA gel, you're going to see something like these two major bands. Okay, now if you are kind of, if you are going to transfer, so this would be your DNA gel electrophoresis image, or sorry, your RNA gel electrophoresis image. What you would do is you would overlay a membrane on top of this and then transfer with an electric current the bands from, or all of the RNA from this gel onto your membrane. Then what you do is you probe that membrane with your radio labeled or your fluorescent probe, and boom, if you see here's a band, or this red line right here corresponds to my probe, and oh, lo and behold, well, yeah, I do see it in RNA sample 1, but maybe I don't see it in RNA sample 2. Is this technique quantitative? Now, the technique itself isn't, as it produces gels with blots. However, with that said, you can make it quantitative, because you can do image analysis. You can look at the density of that particular band. So using an instrument known as a phosphoimager, you can come up with a quantitative measure of that. So to this, I would say ultimately, yes, it is quantitative. Quantitative. All right. Now, uses in medicine. Well, one, it is used for detection of viruses. Now, it's not the most sensitive measure of detection of a virus, but it is something that you, I apologize, that you can do for, yeah, you can detect a virus if that virus is a high enough copy. You can diagnose or correlate gene expression to genetic diseases, and it is ultimately used, or one of the major uses is gene expression analysis and correlating that with cancer biology or with cancers.